Okay. Ever wonder how your body, builds muscle or fights off a virus, even just digests your lunch? It turns out the secret involves these tiny, tiny molecular machines, proteins. Today we're doing a deep dive into them and their building blocks, amino acids. We want to give you a kind of shortcut to understanding these molecules and, well, some of their surprising properties too.
Absolutely. It's pretty amazing how these seemingly simple parts build so much of, well, us. And their behavior is much more dynamic than you might think. By the end of this, you'll have a really solid grasp of their structure and how just small changes in their environment can really impact what they do.
Right. So let's start right at the bottom, these building blocks, amino acids. What exactly is one?
Well, think about all the complexity in us. It really starts here with amino acids. And the amazing thing is there are only 20 common types. Just 20. Each one has a core structure that's basically the same, but then there's this one part, the R-side chain.
Ah, the R group.
Exactly. That R-side chain is what makes each one unique. It gives it its own personality, its properties. So imagine having just 20 types of Lego bricks, but you can build almost anything with them. That's kind of what's happening.
20 types. That's it. Wow. Okay. And I've heard people talk about L and D forms. What's that about? Does it matter?
Oh, it definitely matters. So most amino acids in us, in living things, are the L form. L amino acids. D amino acids, you find them sometimes, maybe bacteria, things like that. It happens because that central carbon atom, the alpha carbon, it's usually asymmetric. Chiral is the technical term.
Like a left and right hand, mirror images.
Precisely, like your hands. They're mirror images, but not superimposable. The only exception, interestingly, is glycine. Its R-side chain is just a single hydrogen atom, so small, which means its alpha carbon isn't chiral, so no L or D form for glycine. It's unique that way.
Okay, got it. So we have these 20 unique building blocks, most with this handedness. But one brick doesn't make a house, right? How do they link up? How do they form something complex, like a protein?
Right. They link up using what's called a peptide bond.
Yeah.
Basically, the carboxyl group, that's one end of an amino acid, links up with the amino group, the other end of the next amino acid. And when this happens, a molecule of water is lost. It's a dehydration reaction.
Okay, so they chain together, losing water.
Exactly. And you get this chain, this linear sequence of amino acids. It has a start, the N-terminal end, and a finish, the C-terminal end. That sequence, that specific order, that's the protein's primary structure. It's like the basic instruction set.
Just the sequence. But proteins have these intricate shapes, don't they?
They absolutely do. That primary sequence is just the beginning. It has to fold. The chain starts to fold locally into simple patterns. You've probably heard of the alpha helix, like a coil. And the beta-pleated sheet, which is more like a folded sheet of paper. This is the secondary structure, local shapes. Then the entire chain, with its helices and sheets, folds up into a very specific overall three-dimensional shape. That's the tertiary structure.
And that's the functional shape.
Usually, yes. For a single polypeptide chain, the tertiary structure is key. It's held together by different kinds of bonds. Some are strong, like bisulfide bonds. Others are weaker. Hydrogen bonds, hydrophobic interactions, electrostatic attractions. Even van der Waals forces play a part. And sometimes you even get multiple folded chains, multiple tertiary structures coming together. That's quaternary structure. Think hemoglobin.
Wow, so it's a whole hierarchy of folding, all starting from that simple chain.
Precisely. And those weak bonds are crucial. They dictate the final precise shape needed for the protein to actually work.
Okay, so these precise structures are vital, but they seem kind of delicate if they rely on weak bonds. What happens if you, say, heat a protein up?
Good question. That leads us to denaturation. Denaturation is when a protein loses its functional shape, its secondary, tertiary, even quaternary structure, because those weak bonds get disrupted. Think about cooking an egg.
Right, the clear part turns white and solid.
Exactly. That's the albumin protein denaturing because of the heat. The heat energy breaks those weak bonds, the protein unfolds and tangles up, and in this case, it's irreversible. It can't go back to being clear liquid albumin. This is why scientists have to be so careful handling proteins. You avoid heat, extreme pH changes, UV light, even shaking too hard sometimes.
Okay, so heat is one factor. You mentioned pH. How does acidity or alkalinity affect them?
Well, amino acids have parts that can gain or lose a proton, which means they can gain or lose an electrical charge. And whether they do depends on the pH of the solution they're in. This is called ionization. And this leads to a really important property, the isoelectric point, or pI.
Isoelectric point.
Yeah, the pI is the specific pH value where the amino acid or the whole protein has an overall net charge of 0. The positive and negative charges balance out perfectly. And here's the key thing. When a protein is at its pI, it's usually least soluble in water. It tends to clump up and precipitate out.
Why is that useful?
Because scientists can exploit this. By adjusting the pH, they can make specific proteins less soluble and separate them from others in a mixture. It's a fundamental technique in biochemistry labs.
Clever. Okay, any other interesting chemical tricks these molecules have? Maybe something we can see?
Definitely. There's light absorption. Some amino acids, the ones with aromatic rings in their R-side chains like phenylalanine, tyrosine, tryptophan, they absorb ultraviolet light really well, especially around 260 to 280 nanometers.
So you can detect them using UV light.
Exactly. You can shine UV light through a solution, measure how much is absorbed, and then tells you how much protein is there. It's a standard way to quantify proteins. And then there's a classic chemical test called the ninhydrin reaction. Yeah. Ninhydrin reacts with free amino groups, those ends of the amino acids we talked about, and it produces this really distinct purple color. What's neat is if you take a protein and break all those peptide bonds using hydrolysis, basically adding water back in, you free up all the individual amino acids. Now you have way more free amino groups available than you did in the intact protein chain. So if you add ninhydrin then, you get a much, much darker purple color. It shows you've broken the protein down.
That makes sense. Okay, so we've covered a lot. Structure, folding, how they react to heat and pH, even how we can detect them. What's the big takeaway here for you listening?
Well, you've seen just how critical these tiny details are, haven't you? The specific sequence, the precise folding, how they interact with their environment. It all underlies, everything, from the food you eat breaking down to your muscles contracting to your immune system working. It's really testament to how elegant and efficient molecular design can be in nature.
It really is a fascinating glimpse into the molecular machinery that makes life possible.
And, it makes you think, if so much depends on these incredibly precise dynamic interactions at the molecular level that we could barely see, what other subtle forces, what other complex interactions are constantly shaping our world, our biology, that we're maybe only just starting to understand?